Next, we have Dr. Choong-Wan Woo, an associate professor at the Department of Biomedical Engineering at Choong-Wan University and an associate director at the Center for Neuroscience Imaging Research at the Institute for Basic Science. Please take the floor, Dr. Woo.
Thank you. So I'm going to talk about promises and challenges in developing neuroimaging biomarkers of pain. So there has been a recent rapid increase in the popularity of neuroimaging biomarker studies. And according to this paper, 6 out of the 10 disease conditions contributing to the global burden are related to neuroscience and neurotechnology. And therefore, non-invasive neuroimaging tools combined with machine learning and AI techniques should play a crucial role in addressing these conditions. And pain is one of the leading causes of disability. And first, let's listen to what Francis Collins, previous NIH director, talked about pain biomarkers.
We need biomarkers that would be able to actually distinguish different kinds of pain in different populations, because the treatment for one kind of pain may be very different than another, and we do too much lumping right now of pain syndromes. And biomarkers that could actually predict clinical outcomes to a particular therapy. We need objective measurements of pain, what we would call a painometer, instead of the very long in place but not all that useful 1 to 10 scale.
There has been a lack of breakthroughs in analgesic development, primarily due to strong placebo responses. And therefore, we need surrogate markers of pain beyond self-report, which is really susceptible to placebo effects. But developing neuroimaging biomarkers of pain is quite challenging because pain involves distributed brain networks. According to this seminal paper, the experience of pain is well correlated with activations across multiple brain regions. This is due to the fact that pain includes multiple subcomponents such as affect, feature extraction, and motor control, and attention, and more. And more recently, Bob Coghill, the same author, proposed that even nociception involves distributed brain systems. In other words, from the very early stage of pain processing noxious stimuli information, there could be multiple brain pathways suggesting the existence of degenerate mechanisms of pain and nociception. Therefore, to effectively model pain in the brain, we need to use systems-level neuroimaging methods such as fMRI and EEG. And that should be the same case for all these neurotechnology targets. And we recently surveyed 57 studies that developed neuroimaging-based pain biomarkers, published between 2009 and 2019. And among these neuroimaging tools, fMRI was the most popular, and with more than half of the studies focusing on clinical populations. And we actually went through and surveyed various aspects of these studies and found the binary classification and population-level models and use of individual-level data and brain-wide data were the most popular approaches. This indicates that researchers who developed the markers of pain are really mindful of the clinical application when they are developing pain biomarkers. But if we examine the use of machine learning models in neuroimaging studies, most of the studies targeted the diagnosis. Then the critical question becomes, will these diagnostic models be clinically useful and informative? And whenever I ask this question to physicians, their responses tend to be quite skeptical. And clinicians, they usually told me that these other difficult questions, such as predicting treatment responses and prognosis or subtyping or identifying risk groups, should be addressed. And this is closely related to the fact that there are many different types of biomarkers. And this is from the BEST resource. BEST stands for biomarkers, endpoints, and other tools produced by FDA NIH Biomarker Working Group. Then which type of biomarkers would be the most useful? And the answer really depends on the clinical conditions and the current status of the understanding of that clinical condition. But more importantly, as a scientist, we might not fully understand the current unmet needs in the clinical field. Therefore, it is really crucial to involve stakeholders early in the process. This means that we need to establish relationships with patients and caregivers and healthcare providers as early as possible. But there are some challenges. The first one, the most important one, is in my opinion, is the poor prediction performance. And this plot is figure 4F from this well-known Marek 2022 Nature paper. It illustrates the linear relationship between univariate and multivariate models of the prediction performance of univariate and multivariate models. However, to me, the key takeaway of this figure is the range of each axis. The performance of both types of models were lower than correlation equals 0.4 across the 41 phenotypes. And additionally, this recent study by Murray Hopley, suggested that predicting individual differences in pain sensitivity may not be feasible. And when this paper came out, I thought, okay, yes, it is indeed difficult to predict individual differences with fMRI, which led us to focus more on within individual predictions. So we decided to investigate this question. What is the maximum accuracy we can get at least for one person? So we launched this My Pain Connectome project, inspired by Russ Poldrek's My Connectome project, but our goal was different. We conducted N=1 pain fMRI study to examine the upper ceiling of the accuracy of fMRI-based prediction of pain and to disentangle population versus individual level brain representations of pain. In this study, we collected two types of data. The first one was MPC1, which involved the extensive sampling of one participant. And the second one is MPC100, which was a cross-sectional study. It involved many participants with a single scan. And the N=1 participant was actually me, because IRB didn't approve of doing extensive sampling with other participants or students at the time. And I underwent 33 sessions, which were 66 hours of scan. And it included 5 sleeping sessions. I intentionally slept through the pain experiment, just out of curiosity, what happens in my brain. And the results I will show you today is based on these 28 awake sessions. And there were two types of stimuli, heat stimulation on the arm and also oral capsaicin. And for MPC100, we collected data from 124 with the same experimental design. And we aimed to predict trial level variations of pain ratings, and we used 3 different modeling inputs. One was the population level prediction based on the MPC100 data set. And the second one was personalized brain model, My Pain Connectome from MPC1 data set. And third was we also included experimental parameters such as stimulus intensity, skin size, and trial orders, etc., to predict trial-by-trial pain rating. And we divided the data into training, validation, and testing data, and all results I will show you today are based on this testing data, independent test data of MPC1. The key results can be summarized with this Venn diagram, which decomposes the explained variance for each model. The total R-squared for modeling trial-by-trial pain rating was 0.684, and R-squared only with the brain was around 0.61. And only with the population brain model, we were able to explain 25.6% of variance. I didn't surprised by this number because this number was so similar to the explained variance that I reported previously. In this paper, I also used the population level model and tried to predict trial-by-trial pain ratings. And with the personalized brain model, the R-squared was 0.605, and the unique variance explained by personalized model was 12.8%. And the variance explained by experimental parameters were 53.5%, which is smaller than personalized model. And also the experimental parameters unique R-squared was also smaller than personalized model unique. So the main point is only with the personalized brain model, we can explain around 60% of trial-by-trial variance in pain ratings. And the personalized model can provide prediction above and beyond the experimental parameters. This provides one answer to a challenge with fMRI, that we could achieve quite high precision prediction performance if we have enough data for each individual. And also, we conducted an extensive sampling study with a small number of patients with fibromyalgia. You can find the detailed results in poster 2507, the Wednesday and Thursday. But then, what about individual differences? Is it really impossible to predict individual differences? And to answer the question, we did the same analysis with hopefully et al. 2022 with MPC 100 data. And with the univariate analysis, we were able to find some regions that showed significant correlations with individual differences in mean pain ratings. In addition, we observed the significant correlations in multivariate pattern-based analysis with the a priori marker and also the regression-based the prediction model. So what brought this difference? Why is this is the case? Our best guess is the sample heterogeneity. So hopefully et al. Intentionally maximize the sample heterogeneity to make their sample more representative of general population. For example, the age range in their paper was 14 to 44 years old, versus ours had a narrower range, 19 to 37 years old. And this interpretation aligns with the results of another paper by Turbo, Clemens, and Marek, which is the response to matters arising piece by Spisak and Torwager here and Bingle. So in their report, they demonstrated higher performance prediction performance with HCP data, which is more homogeneous compared to the ABCD data. But from this, we can conclude that poorer generalization can occur when the sample is heterogeneous, but a larger sample size is always helpful. As you can see, the x-axis increases the sample size, increase the prediction performance. That's always true, I think. But we need to think this through because these two messages might seem contradictory because the larger sample size can increase the heterogeneity in the data. So actually after the long shot, you can think that they have different implications for different sets of data. So top one is related to the testing data, and the bottom one is related to the training set. And then why does the heterogeneous test set reduce the model generalization? And as suggested by this Green et al., another Nature paper, model can fail when it is applied to individuals who do not fit stereotypical profiles. That is, heterogeneity in the test set may increase the likelihood of non-stereotypical individuals. And this phenomenon is because models also reflect the stereotypical profiles, and as well presented in this tweet by the first author, Abigail Green, models systematically fail because they aren't predicting unitary cognitive constructs. They are predicting measurements of these constructs intertwined with the socio-demographic and clinical covariates, stereotypes. I think this is a super important message. And this points us to this important idea that pain is not one thing, and also other cognitive constructs are not the one thing. Rather, pain is complex, integrated brain states intertwined with the socio-demographic and other factors. So thus, therefore, our goal should be to model its component processes, not modeling it as a whole. We can contrast this direct prediction approach with this to the component process modeling approach. In this direct prediction approach on the left, we predict clinical outcomes directly from the brain measures. But in the component process approach, modeling approach, we consider the clinical outcomes to be very complex so that we cannot directly model them directly from the brain. Thus, we first decompose the outcomes into multiple subcomponents and find good models for the component processes. And we can use the analogy of color scale, where the direct prediction approach is a grayscale, whereas the component process modeling approach is RGB scale, so that we can express any colors with them. In this approach, discovering and developing good component targets, intermediate targets, become really important. And this is the last slide. I believe this aligns with the idea of building composite biomarkers, I think which should be a goal in the roadmap for the clinical translation of neuroimaging biomarkers. Thank you for listening. Okay.